Yes, this is Preeti, and I'm second year MBBS student from Hassan Institute of Medical Science. In this video, I'll be saying you few important viva questions from physiology, and I'll be making you understand all these questions so that you remember easily. You need not to mug up anything. First I will say about the neubaurs chamber. Neubaurs chamber has two grids. This is a one grid and this is the other grid. Each grid has nine boxes. Each I'll write these are two grids. These are two grids and each has nine boxes, and each will be this length of this is a square in shape, and this is a 3 millimeter in length, and breadth also 3 millimeters. So each box will be 1 millimeter. Here and here at the four corners you measure WBC. For the WBC count you measure here, and these five boxes 1 2 3 4 5 you will count RBC, where you will measure WBC. This box I am magnifying. Here you will be having 16 smaller boxes. 16 smaller boxes it can be divided into. So this will be 1 millimeter. No, 1 is divided by 4. So 1/4 will be dimension of this thing, and here also 4 1/4. So 1/4 will be 0.25 millimeter will be this thing. The length of this thing will be 0.25, and this RBC square box is further divided into 25 small boxes, 1 2 3 4 5 and then 5. So each 1 millimeter is divided into 1 by 5, so 0.2 millimeter. This is the dimensions and questions about the neubaurs chamber. Neubaurs chamber, this is improved neubaurs chamber, and the depth will be the down will be 1/10. That is 1/10. Depth is 1/10. That is 0.1 millimeter. Improved neubaurs chamber. This is used to measure the count. Is used to count the RBC, number of RBC, number of WBC, and sperm count. For all these three, we use neubaurs chamber. This is one of the question from viva. Another question will be difference between the WBC pipette and RBC pipette. RBC pipette has red bead. You can see red bead, and WBC has white bead. Red blood cells and white blood cells. No, so red bead and white bead. Red bead and white bead, you can easily remember. And volume of this red bead will be larger than volume of this thing, volume of white bulb. This is a red bulb and white bulb. And the tube end will be mouth. This is mouthpiece. Mouthpiece will be red color in red RBC pipette and white color in WBC pipette. Rubber tube will be same. And markings 0.5 initially 0.5 same, 1 and 1 same. Here above this bulb, the marking will be different in WBC pipette because number of RBC cells will be more in millions, but the number of WBC will be only in thousands. So we need not to dilute so much. We can dilute only 11 parts. That is, so the last point is 11 in WBC pipette, whereas in RBC pipette 101. This is the difference between the RBC pipette and WBC pipette, and uses will be RBC pipette used to count RBC count, whereas WBC to total differential leukocyte count or total leukocyte count. For this thing we use WBC pipette. This is a difference between this important question, which is a difference between the RBC pipette and the WBC pipette for the viva. And turks fluid, you need to know the composition of the turks fluid. Composition and the functions of each component of the turks fluid is important question for the viva, physio viva. The composition of turks fluid will be glacial acetic acid, which is 1.5 ml, which is used to a turks fluid. You mainly know that turks fluid is used to count WBC count, so RBC should be lysed, broken down. It lyses RBC. It causes the death of RBC. And the other composition is 1% gelatin violet. That is 1 ml. 1% gelatin ml of 1 ml that stains the nuclei of WBC. Stained WBC nuclei. And distilled water 100 ml as a solvent. This is a composition of turks fluid, and I think you understood the functions of these each component is also. And the other important fluid is for RBC count we use hyaem fluid. Mercury chloride 25 grams, 25 milligrams or 0.25 grams. Sodium sulfate sodium chloride, and distilled water. Distilled water are a solvent. Mercury chloride prevents bacterial or fungal infection. Sodium chloride also. It contains mercury chloride. Mercury chloride acts as a preservative. It preserves from the fungal or bacterial infection. And sodium sulfate. Sodium sulfate prevents the aggregation of RBCs and helps to remain discrete form of RBCs. And sodium chloride. Sodium chloride, it is an electrolyte. It dissolves in Na plus and Cl minus, and it maintains osmolarity. It maintains osmolarity, so it will be isotonic. The solution will be isotonic with the RBC, so it helps in maintaining the size and shape of RBC. And distilled water acts as a solvent. This is all about the heme fluid, which is important question. The composition and the functions of each component will be important for the practical viva question of physiology. And this is all of few practical questions. If you find this video is helpful, please do like, share and subscribe to my channel, and appreciate my efforts.